90,000 people are on a waiting list to get a kidney transplant, and 11 of them die every single day while they wait.
The solution some may find unusual: pigs.
Porcine kidney xenotransplantation represents a turning point by eliminating the organ shortage and offers a solution that is far better than dialysis.
Scientists can use the CRISPR system to edit DNA. So scientists will create a guide RNA and then insert that along with the Cas9 enzyme into the cell's genome. The guide RNA will attach to the DNA target sequence that it is coded for, and then the Cas9 enzyme will cut that target sequence off. Then scientists can use the cell's own machinery to edit the cell's genome.
So, what are we actually doing with CRISPR? Well, there are little sugars called xenoantigens that specific enzymes make inside the pig that we don't have. And if we detect those in the pig transplant, then we're going to reject the organ. Our first example is an enzyme called GGTA1 that produces alpha gal particles inside the pig cells. Since we can't have that, we're going to use CRISPR to knock out the enzyme. That way, we won't have any alpha gal in the kidney transplant. Our next example is a little different, but in essence, it's the same thing. Both humans and pigs have NEU5AC, but pigs have a little thing called CMAH. CMAH transforms this protein into this protein, NEU5DC. Since pigs have this and we don't, we have to get rid of it for the transplant. So we knock out CMAH and then we're done.
Another way that scientists mitigate cross-species viral transmission in xenotransplantation is through inactivating the PERVs found in the pig's genome. PERVs are porcine endogenous retroviruses. Porcine refers to it being from a pig. Endogenous means inside the gene, so these viruses are transmitted vertically and are inherited genetically. Retro refers to how the virus replicates. They use a different method than normal for viral replication. So they will transcribe their RNA and then using a reverse transcriptase convert that into a DNA, and then they will integrate that DNA into the host DNA. And virus refers to it being an infectious agent.
This is how scientists inactivate the PERV sequences in the pig genome. So first, scientists will locate the PERV sequence inside the nucleus of a pig cell. And then, using the CRISPR system mentioned previously, scientists will inactivate that PERV sequence found in the nucleus. Then they will place that DNA into the nucleus of an egg cell using somatic cell nuclear transfer. They will place that egg into a surrogate mother who then births newborn pigs with the inactivated PERVs that can be used in xenotransplantation. The most risky form of PERV that can be transmitted to humans is PERV-AC, which is a result of recombination between the PERV-A virus and the PERV-C virus. It is twice as likely to infect humans as the PERV-A virus on its own.
Another way that scientists use CRISPR to edit pig genes is through adding human genes that make pig tissues less foreign. Imagine if we could solve the organ shortage problem by safely transplanting pig organs into humans. The main problem tends to be in our immune systems because it detects a problem, like a foreign object, and then it tries to destroy it. Some scientists are adding human genes to the pigs in order to combat this problem and blend our biology safely. Specifically, CD59 acts as a shield and blocks the central membrane attack complex. That's a part of our immune system that can actually destroy cells. And with CD59, pig cells get much harder for immune systems to kill. Another key change is adding human thrombomodulin to help with blood clotting and inflammation because pig thrombomodulin doesn't mix well with human blood. But human thrombomodulin actually restores the balance and reduces the rejection rates. Together, the thrombomodulin and the CD59 are genetic upgrades that move us closer to a future where life-saving organs can be grown within animals and bridge that gap between scarcity and survival.